Okay, good afternoon, everybody. A very warm welcome to this webinar, which is part of a series where we're exploring how the engineering biology mission hubs are harnessing engineering biology to address real-world challenges. My name's Pearson. I'm a senior portfolio manager for engineering biology at BBSRC. And in a few moments, I'll be handing over to the Advanced Therapeutics Hub team to talk about the exciting work taking place across their mission hub. But just before I hand over to the team, I want to spend a couple of minutes setting the scene, just in terms of why we consider engineering and biology to be so important at UKRI, and also what we're hoping to achieve through these specific mission hub investments. Small bit of housekeeping to start with. So as this is a webinar, you'll note that your microphone is muted and your camera is turned off. We've also disabled the chat function just to ensure that all your questions are captured in the correct place. And on questions, you're very much encouraged to submit these to the Hub team throughout the webinar. You can do this through the Q&A function. And then all these submitted questions will be collated and posed to the panel at the end of the webinar. And finally, this webinar is being recorded, and we do plan to make the recording available on UKRI's YouTube channel sometime over the next few weeks. So please keep an eye out for that. Okay, so engineering biology is one of our strategic priority areas of investment at UKRI. And the reason for this is because we strongly believe that engineering biology has the potential to deliver huge benefits for the UK in terms of addressing major societal challenges, driving economic growth, and also in increasing national security, resilience and preparedness. As an emerging technology, engineering biology has the potential to transform a broad range of diverse sectors, spanning agriculture and food systems, manufacturing and clean growth, in environmental monitoring and remediation, and also in biomedicine and in improving human health. And it's because of this broad range of applications that engineering biology is a technology that's really recognised globally to be of major importance, with some predicting that it could contribute up to $4 trillion to the global economy over the next 10 or so years. So as part of our strategy to support the UK in realising engineering biology potential, last year, UKRI invested over £100 million in 6 engineering biology mission hubs and in 22 engineering biology mission awards. These investments were made through UKRI's National Engineering Biology Programme, with co-funding support from the wider Technology Missions Fund programme, which has provided a substantial amount of investment to grow UK capabilities in critical technologies. The mission hubs and mission awards build on the UK's world-class engineering biology capabilities to really drive engineering biology to that next level of technology maturity in terms of unlocking its potential across a broad range of application areas and ultimately delivering tangible impacts that make a real difference to how this technology benefits UK society. And now, without further ado, I'm very pleased to introduce one of the Mission Hub project leads, so Professor Susan Rosser from the University of Edinburgh, who, along with her colleagues, is going to explain how the Advanced Therapeutics Hub is harnessing engineering biology to address major challenges in biomedicine. So thank you very much. And with that, I'll pass over to Susan.
Okay, good afternoon, everybody. I hope you can all hear me. So thank you very much for joining us today. What I'm going to tell you about is our engineering biology hub for engineered genetic control systems for advanced therapeutics. So I'm just going to move things forward. So I'm going to start giving you a bit of context to our hub, and then we're going to talk a bit about our approaches and plans. And for that, our colleagues Jane, myself, Sandy and Mark are going to tell us about our actual tool development and approaches. And then I'm going to hand you over to Steve, Giovanni and Alice to give you some example applications of the tools that we've developed. And then Alice will tell you a little bit about at the end about how you can connect with us and work with us in the future. Thank you. So Alice moves on. Okay, so the focus of our hub is how can we use engineering biology for gene therapies? So what is gene therapy? There are a number of ways of thinking about gene therapy. The most obvious one is gene replacement, where we will be replacing genes that have lost their function, they're mutated and they're no longer functional. Another approach is using gene supplementation. This is where you're adding in an additional exogenous gene, and that might be because the endogenous gene doesn't work terribly well, or that you might be adding in a gene that actually mitigates any problems in sort of metabolism, etc. So that would be a supplementation approach. Another example is where you can use RNAi to try and suppress gene regulation, to damp down the protein production, or gene editing, and this is where you actually edit the DNA to change the mutation back to its functional natural DNA sequence, and so that would be using sort of CRISPR tools such as Cas9, etc. And so what we're aiming to do is develop new vectors, and Sandy will be telling you a bit about that later on, and develop new promoters and new therapeutic cargoes. So with gene therapy, you can administer the therapeutic directly into the bloodstream as a systemic and targeted approach, or you can take patient cells out, edit, and use the gene therapies to change the genomics of those cells and then put the cells back into the patient. So gene therapy has been hit in the news a lot recently, and these are 3 examples from the BBC website, recent examples for life-changing therapeutics for leukocyte adhesion deficiency, sickle cell disease, and Leber congenital amaurosis. So there's a lot of excitement about this type of therapeutic. So I've given you a list here of US approved gene therapies. These are some gene therapies and also mutated cells as well. And so there are things coming through the pipeline, and up until 2032, it's expected to be more than 60 product indications coming through, most of them in the oncology space, a lot of them in the orphan indication space, non-oncology space. And so 6% for therapeutics for larger disease areas such as cardiovascular conditions. And we'll touch a little bit on that later on. So next slide, please, Alice. Thank you. So this is an article that was literally from Saturday morning on the BBC. And so in Wales, for example, where I'm from, the government has instigated full genome sequencing for any children who have got an undiagnosed disease. They can't find out any cause of the disease, so they're actually sequencing the full genome of these young kids. And in 40% of those cases, there's a diagnosis of a genetic disease. So there are over 8,000 genetic diseases known, and that's just expanding because as we do more genome sequencing, we're identifying more and more of these genetic diseases which require gene therapy. So the gene therapy market is potentially enormous, looking up to $56 billion by 2034. However, there are problems with gene therapy. There's not as many therapeutics coming through as we would hope. And that is because there are a whole range of problems with gene therapies. One of which is that the vector can cause an immune response. You don't get the delivery to the correct tissue. The transgene can be not switched on enough, so the dosage is too low to be effective, or conversely, the dosage can be too high so that it can cause toxic effects. The gene can be on in the wrong tissue. If you're trying to treat a disease in lungs and it's on, but it's also on in the liver and causing toxicity in the liver, that is not ideal. So the aim of our hub is to use engineering biology approaches to develop a suite of engineering biology tools for gene therapies to make sure that the therapeutic genes are on in the right tissue at the right time and the right level. So who are we? So we're a 40.25 UKRI funded mission hub. Thank you very much to UKRI. We really appreciate your funding. So it's led out of the University of Edinburgh, but with colleagues from the University of Oxford, Imperial College London, and Cancer Research UK Scotland, along with our industrial partners, AstraZeneca, the Cell and Gene Therapy Catapult, Theolytics, BitBio, Lonza, Brooker, etc. And for all you company people out there, we're always open to collaborating more with new partners. So what are we trying to do? So what we're trying to do is try ultimately to benefit patients. So we're trying to build on our scientific and translational excellence. We've got unique facilities, which I'll talk to you about in a minute. And our focus for application spaces on oncology, and that's with our colleagues in CRI UK. They've got fantastic established models in pancreatic, lung and colorectal cancer. I think Steve's going to talk a bit more about oncology later on. Rare diseases, we're focusing primarily initially on lysosomal storage diseases, and we're also using AI for enzyme engineering. With cardiovascular disease, we're also linked quite closely with the £50 million MRC BHF Centre on Heart Repair and Regeneration, which is heart disease, but also vascular diseases. So our hub work packages are, we want to develop a suite of tissue-specific control systems to make sure that the genes are on the correct tissue. We want to be able to control those genes once they're in using small molecule control systems. And we want to develop a whole new suite of viral vectors so that we can deliver our therapeutics to the right tissue appropriately. And as I mentioned, the proof of concept is in oncology, cardiovascular disease and rare diseases. And all of this is underpinned with our social science colleagues in an innovative, responsible research and innovation approach. So for the next few slides, I'm going to tell you a bit about our unique facilities. So this is the Edinburgh Genome Foundry. So the aim is to accelerate biological research and development, providing a fully automated platform for DNA and assembly and phenotyping. The Edinburgh Genome Foundry is probably the most automated, certainly the most automated foundry in Europe, possibly even the world. So on to the next slide, please. So the foundry is genuinely state-of-the-art. We've developed our own software for design of constructs, and then we have a suite of automated systems for putting those DNA pieces together to make our constructs. So we can design our constructs and build them. If you're a postdoc, you can maybe do about 90 odd constructs in a week. With our foundry, we can do up to 2,000 constructs in a week when we run in at full capacity. So I challenge you as listeners to think, if you had the capacity to build 2,000 genetic constructs, what would you do with that? So along with our ability to build DNA, we also have a unique facility to phenotype DNA, to phenotype cells that we put those constructs into. So this is a Bruker beacon system, which is the only one in academic hands in Europe. And we can do a whole suite of assays on this piece of equipment. So let's go on to the next slide. So how this works is we can take single cells and use light to take individual cells into these nanopens. In the nanopens, we can do a range of assays, identify the individual single cells that have the phenotype that we want, and then use a light ladder to take the cells out of the nanopens and take them off to a 96-well place. So we can look at a whole population, but look at individual cells, phenotype them, and then take those cells back and do further work on them. So the kind of thing that we can use this approach for is antibody discovery. So the diagram at the bottom shows a workflow. So we can go from mouse or human B cells and actually identify antibodies to a specific antigen in a matter of hours. So this is an amazing capacity. So how it works is you have the antigen attached to beads which flow across the top of the nanopens, and you use an assay using a fluorescent marker to identify the pens that contain B cells producing the antibody of choice that is interested, the antibody that links to the antigen on the beads, and you get these little blooms that identify the wells that have got the cells of interest in. The other assay that we've just literally just started working on is a CAR T cell assay. And in this instance, you can put your CAR T cells in the pens with tumour cells and do a whole suite of assays such as cytokine secretion, a T cell proliferation assay, and a cytotoxicity assay. You can actually watch the T cells killing the tumour cells. And again, you can export the cells, the T cells of interest, and get those back to do future work on. So I would encourage listeners to think about how you could utilise these kind of assays in your own research. Next slide. As part of the funding for the hub, we're developing a new automated mammalian cell culture platform, which should be up and running in about a year's time. So we'd be keen to work with colleagues on mammalian cell engineering using a Foundry, our Beacon, and our mammalian cell culture system. I'm now going to pass you over to Jane, who's going to tell you something about the RRI component.
Hi, thanks very much. So I'm just going to talk very briefly about the responsible research and innovation component of the hub. Our aim is to collaborate closely with other work packages to explore the social, political and ethical dimensions of the research in the hub, and also to develop capacity for responsible research and innovation in the hub and beyond. And this is because advanced therapeutics could radically disrupt existing models of medicine, and RRI helps ensure that what we develop is equitable and in line with the public interest. And our work is driven by the question, what kind of societies and futures are we building with our technologies? So there are 4 of us on the team, myself, Rob Smith, and the postdocs Ruben Message and Sophie Stone. We're all based in science, technology and innovation studies at Edinburgh. And our work is based on over 15 years of engagement between social scientists and synthetic biologists at Edinburgh and beyond. Next slide, please. So the 3 topics that we're going to focus on is firstly exploring how the hub is developing tools and how this connects with their anticipated applications. Secondly, looking towards applications by building a collective understanding of efforts to scale up and commercialize at the university industry interface. And finally, broadening engagement to publics and stakeholders to investigate understandings of advanced therapeutics and future research priorities. And we hope that building knowledge across all these topics will be really important in ensuring long-term integration of the hub's technologies into both industrial and healthcare landscapes. Next slide, please. So the methodological approaches we will use are collaboration, space, and time. And our work will involve fostering collaborations between a diverse range of disciplines and audiences and stakeholders, exploring the spaces in which the hub's work takes place to learn about how to innovate responsibly, and importantly, creating space and time for these types of discussion, which is quite difficult when we're also busy. And finally, analysing the ways in which an engineering biology approach to advanced therapeutics particularly brings up issues concerning space, time and scale. For example, the manufacturing models used, the geographical distribution of the engineering biology capabilities, and the pace of the work that's done.
Okay, so for the next few slides, we're going to start talking about what are the tools that we're beginning to develop. So we need to deliver our therapeutics to the right tissue, and Sandy's going to start talk to us about that. We also want the genes to be switched on in the right tissue, so using synthetic specific promoter enhancer sequences, and Steve will tell us a bit about that later on. And another method that we can use to look at tissue specificity is a splice variant. So we're going to be looking at sequencing different tissues and looking at the difference in splicing between those tissues, and we can use those splice variants to generate tools that enable us to switch genes on very much in the specific tissue. So the diagram shows some types of alternative splicing, and we're going to be looking at, as I said, at tissue specificity. We will also be looking at using small molecule inducers of splicing, and I'll talk a little bit about that in a couple of slides' time. Next slide, thank you. So what we aim to do is to take healthy tissue organoids and organoids that are from cancer patients, and these are healthy tissue and tumour tissue from the same patient, do a long-read sequencing, RNA-seq, and identify different splice variants. We can then engineer tools that build on this so that the start codons are only present in the splice variant for the tumour tissue and not in the healthy tissue. And by using that approach, we can build genetic tools that only enable a gene to be switched on in the tumour tissue, but not in the healthy tissue. Next slide, please, Alice. And so it's known from the literature that small molecules can also induce specific splicing. So we've been taking cells and applying inducer molecules. So we can then do sequencing plus and minus inducer molecules, and again identify particular splicing effects when a drug is applied, but which are different from when there's no drug present. And so we've got 4 drugs on the go at the minute that induce splicing and are building these genetic switches based on splicing induced by these inducer molecules. And we've just started looking at some RT-PCR, and we're seeing splicing. So we've just started doing that, and now we're going to build on that using different inducers. Next slide. So the other approach that we're using to control transgenes is using aptazymes. So this is control at the RNA level. So aptazymes are small RNA sequences which have a particular 3-dimensional structure, and then they can bind small molecules. And when they bind small molecules, you can choose whether you have an on or an off switch. So the binding will either enhance the RNA breakdown or prevent it. So in this instance, we're using as an exemplar small molecule caffeine. So the aptazyme binds to the caffeine and prevents the RNA being cut, and so you get GFP being produced. And if we've used the beacon system to identify these aptazymes which have got good switching properties. So if you look at the video on the right hand side, what's happening there is we've got cells with the aptazyme in them, and so you're getting GFP produced. So when caffeine is present, we flow caffeine in, the GFP is produced, and when caffeine is removed, the GFP goes away. So we've got a switch that uses a small molecule control, enabling protein production. So caffeine clearly is not going to be the best molecule for a therapeutic, but we have aptazymes being produced for other small molecules which are FDA approved. They've been used in the clinic, and they are far more appropriate for therapeutic use. Next slide, thank you. So in this approach, we want to be able to control how much of a transgene is produced. And as an example, this is the disease Rett syndrome, which is a neurodevelopmental disorder. And the mutation is in the gene MECP2. And so with MECP2, if there's a mutation in it and you're not producing enough MECP2, then you get neurodevelopment disorders. However, you can get an MECP2 duplication syndrome as well. And that's where there's too much MECP2, and that also results in neurodevelopmental disorders. So if you're producing a gene therapy application, then what you want to be able to do is have the transgene expressed at a therapeutic level. And so in order to do this, in collaboration with the lab of Stuart Cobb, we produced a synthetic system where we have a microRNA binding site. So microRNA is produced, and then that can control the level of transgene expression. So it's at a therapeutic dose. This has actually been it was tried in mice, and you can see the data from the mice on the right-hand side, lower right-hand side there. It's been patented. It's been licensed to a company, Neurogene. This was taken into non-human primates, where it's also proved effective, and it's now in phase 1, phase 2 clinical trials. So with Diego, we are also, and also Giovanni will speak later on, we're leveraging the power of AI and machine learning, and we're looking at how we can use these approaches to predict RNA designs for our aptamers and synthetic promoters, and just general control, precise control of our gene therapeutics using these AI machine learning approaches. I'm now going to hand you over to Sandy, who's going to talk about viral vectors.
Thanks very much, Susan. So I'm leading a team of a number of groups in the University of Oxford contributing to the hub. And we're contributing in the broad area of viral vectors for delivery of all the new construct technologies, control technologies being developed by other hub members. So of the different groups between us, we're covering 3 different viral technologies. Adenovirus in my group, and then I'll tell you on subsequent slides about AAV, adeno-associated virus, and lentiviral based approaches. So adenovirus, as many people will be aware, were the platform technology for the Oxford AZ COVID vaccine. They've also been used in oncolytics, and in the past have been used, were explored quite extensively for gene therapy, but then faded away from that field. They do, however, have important advantages and we think considerable potential for gene therapy, notably their large transgene capacity. And so this project is going to work primarily upon seeing if we can target adenoviruses to tissues, especially respiratory mucosa, more effectively than current versions. Next slide, please, Alice. So Erwan Pollard, who's a very talented postdoctoral scientist in my team, has developed technology for directed evolution in adenoviruses, and we hope that this will bring to the adenoviral field more powerful library selection technologies that have previously been successful in the AAV and lenti field, but have been hindered from contributing an adenovirus by molecular biology hurdles, essentially. Next slide, please. And so Erwan plans to generate libraries of adenovirus vectors aiming for improved tissue tropism properties, and then bioselect them in ex vivo human models to select vectors with the properties we're interested in. Next slide, please. Work in Kerry Fisher's lab is going to be working on adeno-associated viruses. And as many people will be aware, they're already extensively used in licensed clinical products, but there remain important challenges and limitations, notably neutralizing antibody and difficulty in targeting certain target tissues. And so Kerry is planning to apply capsid engineering to overcome those challenges, particularly with a view to targeting colorectal cancer metastasis. Next slide, please. So Kerry's work will be taking advantage of 2 real technical advances. So one of them is in the construction of the libraries themselves, using an adenovirus-based AAV production technology, which his colleague Len Seymour developed in the past. And the other is around ex vivo perfusion of human organs. And in this case, this is human livers from cancer resection, so liver specimens containing actual colorectal cancer metastases. And he will be using that as a platform to bioselect AAV vectors with the potential to transduce these tumors. Next slide, please. So this is just an overview of Kerry's project, as I discussed. So bringing together that library creation technology with the ex vivo human liver perfusion model, and we hope working also with the other groups in the hub, in particular in Scotland, with the cancer organoid experience. Next slide, please. And then the final group contributing on the Oxford side of this project is Steve Hyde and Deborah Gill's group. Some people will be aware they've got extensive experience of lentivirus-based gene therapy over the years, and they're particularly interested in targeting lung for rare lung diseases. So one of the 2 projects in their group will be using lentivirus-derived nanoparticles, which is a new approach to payload delivery. So from the outside, these look like a lentivirus. They can be pseudotyped in the same way as a lentivirus, but rather than packaging a vector genome, these LVNPs instead package a protein or a ribonucleoprotein, such as a Cas9 based or base editor or prime editor editing payload. So these will allow the strengths of lentiviruses to be combined with the power of CRISPR-Cas based genome editing. If you just advance the slide again, Alice. So they will be planning to exemplify this for a rare disease target, a surfactant protein C dysfunction. and investigating different genome engineering formats for the payload. Next slide, please. And then the second project in Steve and Deborah's group will be looking still within the area of targeting rare lung disease. They will be looking at drug-inducible cell-specific transgene expression switches. So they have some preliminary data using rapamycin analog and a humanized protein dimerization system. And this is potentially applicable both to lentivirus delivery and also to AAV. They plan to take what they have at the moment and turn that into a tissue-specific switch system. Next slide, please. And I think at this point I'm handing over to Mark at Imperial.
Yes, thanks, Andy. So at Imperial, we're contributing to two aspects of the Mission Hub. First of all, in AAV adeno-associated vectors, much like what Sandy described, and also in payloads to go with them. I'll begin by presenting the work of Professor Jeff Baldwin's laboratory, who, within the context of the Hub, is developing AAVs to be expressed rather than in their standard form in HEC293 mammalian cells to make a new kind of AAV that is expressed more efficiently within E. coli bacterial cells. And the reason for that is that AAVs are particularly useful in gene therapies. We counted over 130 human clinical trials using AAVs in the last few years, and several products, at least 3 approved by the FDA for human use already. But the problem is that the production with these HEC293 T cells, it's very slow, it's very expensive, and it would be really advantageous if some of the common AAV tropisms, such as AAV9, could be converted to a really scalable, easy to use bacterial expression platform. So Jeff Baldwin's group is working on that by looking at the viral proteins that you'd need minimally in order to make AAVs in the lab. Could we have the next slide, please? And Jeff is using a special method of DNA assembly that's particularly suited to high throughput biofoundry type applications. It's called BASIC, and it's a standardized form of cloning that allows you to make tens, hundreds, even thousands of constructs very, very easily. And so this is used to shuffle genetic parts together and build lots of plasmids in order to test all the different variants and levels of expression that you might need in order to make an AAV9 expressed properly in E. coli. Next slide, please. Towards that, one assay that Jeff's lab has developed is using GFP as a marker for correct folding of the viral capsid particles. In this example that you can see on this slide, there is the viral protein 3 that's fused to GFP, and only if the VP3 folds and assembles correctly do you get levels of green fluorescence, and this level of fluorescence is correlated to the amount of correctly folded production. So it's essentially a proxy for an assembly efficiency test in vitro in order to see whether your particular one in many constructs is making a good level of protein expression. So in the initial screens that they are doing in Jeff's lab, they're testing more than 20 genetic combinations with different linkers, different expression strengths, etc., in order to begin to piece together these viral capsid-like constructs. Next slide, please. So once you have got an AAV that you're able to make, what are you going to put in it? And that's what we are working on in my own group. We're working on the payloads and particularly genetic switches that we can deliver using AAVs. We've been working with small protein transcription factors that we can design to bind to pretty much any DNA sequence we like for many years now. And we have delivered them already in vivo, showing that we can upregulate targets, downregulate targets for many months in quite challenging tissues such as brain, using just a single injection to deliver the AAV via various delivery routes. But this is still a problem in terms of the fact that AAV delivery is very expensive. When you give large doses in order to get the full efficiency, you can end up with getting toxicity in human patients. And so we're looking for ways to really reduce the dosage of AAV that's required in order to deliver our little gene regulatory transcription factors that we call these SYN-TFs, synthetic TFs that bind and target any specific DNA target we want. And so in order to overcome this dosage problem, we've come up with this framework of bystander cell delivery, where we have used engineering biology to put little modules onto our synthetic transcription factors, first of all so that they're secreted from the AAV infected cells, then the secretion signal is cleaved off, and then they reveal penetration signals to penetrate quite efficiently into neighboring cells. And what this means is that you don't have to hit every single cell in your target tissue in order to regulate gene expression. So for example, in a tumor, it wouldn't matter if you don't get every single cell in your tumor first time round with your AAV vector. Once you transduce some cells, you will start to secrete and cell penetrate to all the neighbors, really amplifying the dose. So this enables you to work with a much lower effective dose. And we've already shown with other non-cancer targets so far that we can achieve this quite efficiently in humans, including in, sorry, not in humans, in mammals, including in the brain and in muscle. Next slide, please. Our payloads are really one aspect that we're working on a lot. So for this consortium, we're developing synthetic transcription factors against a number of cancer targets in order to provide essentially genetic switches, mainly to switch off oncogenes, but also we have the potential to switch them back on. The technology is based on zinc fingers, which are natural human DNA binding domains. This means they're very immunocompatible and you can express them for a long time inside a mammal without having any immune reaction. On the left-hand side, you can see a schematic of how one of these proteins can be re-engineered to bind any short DNA sequence you like. So the 3 X's you see there are 3 DNA bases. And by changing the protein positions in these numbered circles, -1, 2, 3, and 6, you can essentially specify any DNA sequence you want to bind to according to an amino acid code, which you see on the right-hand side. So by changing on the right-hand side position -1 and putting a particular amino acid in that position, you can specify whether to bind the DNA base G, A, T, or C. And then you can make long chains of these, so you can bind quite long targets. We typically make chains of these zinc fingers to bind 20 to 30 base pairs, which is sufficient to get a single specific hit in the human genome. So we could target an oncogene specifically using this kind of technology. So then if we move on, we already have some preliminary data where we've looked at a few cancer genes, and we've developed fluorescence assays as part of this particular Mission Hub consortium, where green fluorescence, D2-EGFP on the y-axis of these graphs, shows you whether you can repress a target DNA that's associated with a cancer gene target that's specific within the human genome. So the examples you can see here in the red rectangle boxes are examples with and without the DNA binding domain to repress the target gene. And you can see that without repression, you get a high bar. And once you add your treatment, you can repress up to around 80% your target gene. So we can make many of these. It's really scalable. We have a platform, and in theory, we could make tens or even hundreds of these little payloads against all kinds of different cancer targets. And so then we're really excited about using this within the oncology space. So I will hand over because I think the next talk is really about oncology.
Great, thanks very much. Thanks very much, Mark. So I'm Steve Pollard, University of Edinburgh, in the Cancer Research UK Scotland Centre. So I'm going to just give a few slides on the exemplars of first brain tumours, but some other solid tumours where these types of technologies could be really important. So my own area of interest and where we come into this was through work on glioblastoma, the most common and aggressive primary brain tumour. In previous synthetic biology centre funded work here in Edinburgh, we developed what we call synthetic super enhancer switches that are able to control payloads very exquisitely. That led to the formation of a spin-out company, Trigenics, and a spin-out from the University of Edinburgh, where we get the precision, finally the precision to control transgene expression. We use AAV vectors because they're a fantastic sort of Trojan horse approach to deliver effectively. And then the goal is to kill the cancer cells we transduce, but to train and educate the immune system. And so we had some really nice preclinical data from that, which I'll show next, and that's moving to clinic in the next within the next year. So just one little piece of data here. This is using AAV with a synthetic super enhancer switch. We have a kill with a bystander and then a cytokine-delivered immunotherapy. And so when we inject this into brain tumors, shown here in the heat map, established brain tumors that are going to kill that mouse within a week or two, we see tumors melt away and disappear, and the mice are fine for a year or more. So we have some exciting sort of steps forward here, but we know we need to go much further in the future and broaden this out. Next slide, please. So with that in mind, the core work packages that will be involved are summarized here. So what I just showed you is the top panel here with A and B, 2 payloads from a promoter with an AAV. And what we would like to do is to get much more cancer selectivity. We would get much better, would like to get much better immunotherapy payloads, and we would like to disrupt some of the gene regulatory networks that drive the cancer. So looking for, as Susan said right at the start, how do we get a really high dose, but in a controlled, precise way? And there are really 4 areas shown in the yellow box here that we go after. The first is CRISPR payloads, identifying loci in the cancer genome that are vulnerable to cleavage. To go after tumor suppressors and novel minimal versions of those, which may be effective in AAV vector delivery. Nanobodies and antibodies are an obvious place for us to go, but to use those in combinations for the first time in vectorized antibodies. And also some projects in microbial proteins, which have the advantage of potentially disrupting the chromatin state, but also being highly immunogenic. So it's really identifying individual novel factors here. At the top, the individual payloads, but then screening and delivering those at scale in optimal combinations with the optimal transgene architecture. And you would have seen from all the slides so far, the ability to sort of plug and play different capsids, different control elements, DNA, RNA-based, different combinations, using different repertoires of payloads will be, I think, really powerful. So the next slide just shows you the line of sight for other tumor types. These are all solid tumors shown on the top right here, solid tumors of unmet need, where there's a desperate need for novel killing of the tumor, but also the immunotherapy approaches we have at the moment are not working that well. So glioblastoma, liver cancers, so hepatocellular carcinoma and liver metastases, but also pancreatic cancer. And we're fortunate with Owen Sanson and the CRUK Scotland Institute, former Pearson Institute. We have a real wealth of models, both mouse models and human organoid models, which Susan mentioned earlier. So those are the tumor types we will focus on. Next slide and last slide from me is on the pancreatic cancer models, which come from the Glasgow team, Jen Morton and others, and just gives you a feeling for the types of models they have there that are really gold standard. They recapitulate disease histopathology, disease biology, and importantly for what we're proposing here, the syngeneic. So you have a immune competent scenario where those tumors are killing the mouse by somehow evading immune destruction. So I'll maybe stop there and pass over, but that's the quick summary of the cancer programs.
Yeah, thanks, Stephen. As a way to exploit the power of gene therapies is to treat the monogenic diseases, and my group here in Edinburgh is interested in developing new therapies for lysosomal storage diseases. Next. These are diseases that are caused by inherited mutations that inactivate lysosomal enzymes in each and every cell. And what happens is that you get toxic accumulations of lipids, glycoproteins, and molecular polysaccharides in each one of them, causing cellular death and ultimately tissue and organ damage and early mortality. So these are rare diseases. There's a broad prevalence as a whole, ranking from 1 in 5,000 and 1 in 100,000. But this is the reality is that there's an increasing prevalence and incidence thanks to neonatal genetic screening. And once you get diagnosed, this is chronic conditions that affect the lifespan of the individual. So it gets diagnosed in kids and adult, and you are treated for the rest of your life. And as you would expect, when something doesn't work, you wanna replace the missing and broken piece with something that kind of give you back the missing functions, and that's the idea behind enzyme replacement therapies, which are the standard of care for most LSDs. It's a well-established delivery method of these recombinant enzymes made in the lab that allow us to clear out the substrates in each cell. However, there is a varying efficacy, which depends on both the deficient enzyme and the affected organism. And of course, among the challenges that we have at the therapeutic level is the usual poor stability and poor activity of these enzymes, which means that the patients go through weekly or biweekly injections of these therapies to prevent the accumulation of the substrates. They are usually highly immunogenic, depending on the mode of inheritance of the specific disease, and which means that the patients develop anti-drug antibodies that pretty much stop the efficacy of any ERT, and usually as these enzymes are pretty big, it's difficult to deliver to many organs, especially the brain, due to the blood-brain barrier. So the idea is that we want to change the next slide. We want to change the amino acid sequence of the protein to try to preserve the function of the enzyme, but obtaining better therapeutic properties. And the way we do this is embracing the engineering biology mantra of design, build, test, learn cycle. And through the years, we have developed this platform that stems from our artificial intelligence model that is able to redesign proteins starting from protein sequences and identify potential beneficial mutations, which then are prioritized and brought to our labs, where we have an high throughput screening platform based on Pichia pastoris. It's yeast largely used in the biotechnology industry, including pharmaceutical ones, which allow us to identify lead candidates in as little as 2 weeks. And once we have the lead candidates, what we do is we make a near medical grade product by expressing these enzymes in Chinese hamster ovary cell lines, which is the standard de facto for this industry. And of course, for each enzyme, we have a battery of automated systems to characterize their stability, catalytic activity, immunogenicity, and resistance to changing temperatures. And all this information is feedback to our AI model to improve further our proteins with the aim of obtaining the best possible therapeutics as well. So we try to move ahead from the ERT limitations by developing gene therapies for lysosomal diseases that can be delivered effectively to any tissues. And we are going to focus here on 2 diseases, particularly Fabry disease, which is caused by deficiency of the human alpha-galactosidase enzyme, is one of the most, if not the most, prevalent lysosomal storage disease. All the current treatments are ineffective because of poor enzymatic stability, but also for males, the high immunogenicity of the current offering. And we already, as part of the hub and the previous project, we have identified the lead candidates that we plan to put into AAV vectors to make sure that we can deliver these enzymes to every cell and have a stable expression, thus reducing any recurrent administration of this therapy. And the other one that we are tackling as part of the hub is Morquio syndrome. Is an ultra-rare disease affecting on average 1 in 100,000 people. It's mostly affecting the bones, which are notoriously a difficult tissue to target. And that's why we want to turn into the gene therapy to provide really patients with a viable treatment. And we know already that this is the most expensive therapy out there. It's an ERT that has no effect. And so there's a pressing need to come up with better therapeutic options for the people affected by this. And I think I'll pass to Alice now. Thank you.
Hi everyone, I'm Alice Bailly. I'm the head of operations for the hub. We're running slightly behind our schedule, so I'll run through just one of the last examples that we've got about how we can apply gene therapy to cardiovascular diseases before I wrap up the webinar. So this is actually work from Andy Baker's lab at the University of Edinburgh, and the application here is about a surgery. So what happens is if you've got atherosclerosis, so your arteries can get blocked because of a buildup of fat or cholesterol, and if it's quite severe, you're at risk of cardiac arrest. So the surgical measure is you take a vein from your leg and you replace your diseased artery with that vein. It's quite a common procedure. It's done on a regular basis in the UK, but the challenge here is within a 5-10 years window, you've got about half of your veins that get blocked again. And the reason it gets blocked again is a slightly different process. It's just because of the way that we're because of the fact cutting that vein in the leg and putting it in a heart. The vein was not quite designed to do this in the first place. Plus, the act of cutting the head causes inflammation, and what happens is there's a specific type of cells in that vein that starts to proliferate, and it will eventually create a blockage again. So you're back to square one, and the person may require surgery again. So how are we planning to use gene therapy here? So the plan is to target especially those cells that proliferate and cause those blockages. So they're called smooth muscle cells. And the beauty here is because you're in surgery and you've got access to that vein before it gets put back in the patient's body, and so you can use that kind of time window, about half an hour, to apply some gene therapy. And the idea is to make it as specific to target to make sure that those cells won't proliferate once they're in the patient's body. So that's what the hub work is about for Andy's lab, just making sure it's as specific as possible, and will prevent that proliferation of cells and make sure that if you get surgery the first time around, it will work in the long term. So just to wrap up very quickly, so we've got time for Q&A. So back to our mission. So Susan mentioned quite a few positive success stories, and if I in a nutshell, that's what our hub is all about. We want more of those success stories on gene therapies going forward, and we've got a commitment within the hub really to get things to the patients. That's something that we care deeply about. We've got lots of expertise within our hub of people who have that experience of taking things into the clinic. And there's also a piece of work within the hub, also thanks to UKRI's seeds funding, to really help create a translational culture. And by this is making sure that people in our hub have the skills, the connections, the expertise to take things to the patients. So I'll just wade through because of time. But in a nutshell, while there are things that we can do ourselves, impactful science rarely happens on its own. So we're very much open and desire collaborations. That's how we do the best of work, really. So if you're there in the audience, or you know someone else who's not there but can be signposted to us, we're very much open to collaborating with them if you share our mission and delivery. So and collaboration with the hub can take many forms. It can be academic collaboration. It can be collaboration with industry partners, which we have experience with, and think are necessary here to get things to the patient. Can take many different forms. So if you please do connect with us, just come on board, really. If you think you can help us accelerate that science to get to the patient, we want to hear from you. So there are a few ways to get in touch with us. You can email us at the email address that appears on your screen. You can also find us on LinkedIn and even on Blue Sky. There are the QR codes here. If you scan them, it should take us to your web pages, and you're always free to just get in touch directly with any of us as you wish. So that's it for me. I'm conscious we've got 4 minutes to Q&A, and I'll hand over back to Tom.
Okay, thanks very much, everybody. We're going to move on to a Q&A session now. We do have a few minutes for questions, so please do start to submit those now. And perhaps while we're waiting for some, I might use my prerogative as chair to ask one question. So you mentioned a few times that you were kind of open to engagement with additional industrial partners. And I was wondering if you could kind of expand on what sort of specific areas of the hub, so particular kind of science areas that you'd be particularly interested in collaborating with industry in, or that industry might be particularly excited to kind of work with you on.
So we're open to all areas. So all of our colleagues would be delighted to work with industry. I think that in discussions with industrial collaborators, the ability to control gene expression seems to excite all of them for different applications. And it's not even just in the gene therapy space, but in the manufacturing space as well. So if you can control gene expression, then that also has a big impact on production of viral vectors, for example. Even production of monoclonal antibodies, for example. So all sorts of engineering biology control systems seem to be of interest to our industrial collaborators. They also seem to be very interested in access to disease models, because I think that's something that they find quite challenging to get access to. And I think that all people we've spoken to in industry are always very keen on any new therapeutics that are coming down the pipeline, because they all want to expand their pipelines. And yeah, as I said, we're very open to have a chat and discuss industry-specific problems and talk to them. I think that the collaboration works best when you have an open and honest dialogue, and they can tell us what their problems are, and we can tell them what our problems are, because often we're unaware of the issues that exist in industry. So it's that open discussion is where things become most valuable.
Can I also add, so the hub is very much focused on gene therapies, but we are very much in the advanced therapeutic space, and so and a lot of what people are doing can be applied to cell therapy. So yeah. It's really about advanced therapeutics, even though most of the work that we are doing just now is focused on gene therapies.
Yeah, I would absolutely reiterate that. I think a lot of the applications and the approaches can equally be applied to cell therapies. I think as well, I've talked a bit about the facilities that we have here at Edinburgh with the Genome Foundry. The Genome Foundry is very open to collaboration with academic colleagues and industrial colleagues. So if some of the equipment that we've got is of interest to you, then please get in touch. I know that Oxford has also got a whole suite of fantastic facilities, which we didn't have time to discuss today, and I'm sure that they would be very open and interested in collaborating with academic and industrial colleagues on use of their facilities as well.
Okay, thank you very much. So I think unfortunately we're nearly out of time. So perhaps the last word could just be if people wanted to get in touch with you to ask questions or indeed to explore collaborations, could you just repeat what the best way to do that is?
So you can email either engbiomissionhub@ed.ac.uk. You can find us on LinkedIn, or you can even just find myself directly. So that's Alice Barrier, alice.barrier@ed.ac.uk. And anyone else in the hub will signpost you. So it's not just Edinburgh, it's across all the organisation. We'll get you to the right person. But yeah, if you contact me in the first instance, I will get back to you and we'll take it from there. It always starts with a conversation for science to make impact. So.
Yeah, or equally, if you can just contact me directly, I'm pretty easy to find on the internet. I'm very happy to discuss.
Brilliant. Well, thank you very much to the Advanced Therapeutics Hub for the excellent presentation today. And thanks very much to all the attendees for coming along.